Alright, so I'm going to talk about meiosis and the evolution of sexual reproduction. Here are your learning targets for the week, including explaining the relationship between DNA, genes, alleles, and chromosomes. Later this week, you're going to draw and label a diagram that shows the stages of meiosis, and I'll talk about those stages of meiosis in this PowerPoint. You're going to compare oogenesis and spermatogenesis, so basically the creation of egg cells with the creation of sperm cells. You're going to compare sexual and asexual reproduction, and we're also going to talk about the advantage of sexual reproduction in terms of how it can lead to natural selection. So starting here with chromosomes. Chromosomes are long segments of DNA, and we've already talked about the structure of a chromosome, how it's wrapped around histone proteins. But what you should know is that one long stretch of DNA, which makes up one single chromosome, depending on how long it is, could have between several hundred and a few thousand genes. So genes are one part on one chromosome. What we're looking at in this picture is something called a karyotype, which is basically an image of chromosomes that was taken when the chromosomes were in late prophase, so at the very, very beginning of mitosis. And if you remember, this is when we can actually see chromosomes because they have to condense and get real tight and coiled in order to be able to organize them and separate them from each other during meiosis and end mitosis during cell division. One term that's really important that you understand for this unit is this term right here, homologous pairs. So what we're looking at here is a karyotype. It's a picture. It was taken from somebody's cells, and they kind of cut out the chromosomes and then organized them by size. And so the largest ones are here, and the smallest ones are here, and then right here are the sex chromosomes, which determine the biological gender of the individual. And so this individual has 2 of chromosome number 1, 2 of chromosome number 2, 2 of chromosome number 3, and that's how it works for each of their chromosomes. They have 2. Now the reason why we have 2 of each is because we belong to a sexually reproducing species, which means that we inherited one of these chromosomes from our mother and one of these chromosomes from our father. And so one of these is the paternal chromosome and the other is the maternal chromosome. So they have, the reason why we call them homologous is because they are similar to each other, right? Homo means similar or same. But what you should understand about homologous chromosomes is that they are not identical to each other. So in other words, yes, this chromosome is the same size as this one, and they even have the same genes on them. But this one might have a slightly different version of a particular gene than this one, because this individual inherited a different version of a gene from her mother than she did from her father. And so homologous means that they're similar to each other but not the same. All right, and another thing to just point out about these pictures is that remember that the chromosomes duplicate during the S phase of the cell cycle of interphase. And we are therefore looking at duplicated chromosomes. Even though you can't see the second half of it, this right here is one single duplicated chromosome, and this is one single duplicated chromosome as well. So it's hard to see that there's 2 halves to it, but there are 2 halves to those chromosomes. Those are in their duplicated state. All right, so genes and alleles. So I already mentioned that chromosomes can have between hundreds and thousands of genes on them depending on the size of the chromosome. And so a gene, therefore, is a section on an organism's DNA that holds the information for a particular trait. And remember, we talked about proteins are what determine those traits. So therefore, genes hold the information for making proteins. So in a sexually reproducing species, individuals get pairs of genes because they get one chromosome from their mother and then they get a second chromosome from their father that are going to have the same genes on them. So one maternal chromosome and one paternal chromosome. And the genes here are the same as the genes here, but you might have slightly different versions of those genes. Now, we have a word for different versions of genes, and that word is alleles. So alleles are different forms of the same gene. And we have an example down here of homologous chromosomes inherited by a plant. And here's the chromosome from one of the parents. Let's say it's from the mother. And here's the chromosome from the other parent. Let's say that's the father. And so the gene for flower color for this plant is located in this specific place. Its locus, locus is the word that means location on a gene, is right here. And that means the location or the locus on the homologous chromosome is going to be in the same place. And this parent had a slightly different version of flower color, right? So they both hold information on these chromosomes for determining the color of the flowers. But what's different is what color it's going to end up being. In this case, the parent has an allele for purple, and this parent had an allele for white. And so now this individual has two different alleles for the same gene, the purple allele and the white allele. And that's how we are for almost all of our genes. We're going to get two different alleles for all of our genes because we'll get one from our mother and one from our father. Now, I mentioned almost all because there are some exceptions to that, especially in males that only have one X chromosome. All right, so meiosis is very similar to the process of mitosis, which we just learned about. And this is the process in which an individual's genome is reduced from two sets of chromosomes. So the symbol we use for this is 2n. We call this the diploid number to one set of chromosomes, or n, which is also called the haploid number. And so these chromosomes are isolated into special cells that are called gametes. In flowers, right, flowering plants, the gametes are found in these specialized organs. This is called an anther, where the male gametes are formed, and this is called the ovary, which is where the female gametes are formed. In animals, our gonadal tissue is where the gametes are formed, and in different places depending on if you're male or female. Almost all of our cells in our bodies are diploid cells, and they're going to have two non-identical copies of nearly every chromosome. And that's, as I said before, because we get a copy of all of our chromosomes from our mother, and then another copy of each of those chromosomes from our father. But the difference between females and males is that females have two X chromosomes, so they inherited one X from their mother and one X from their father, whereas males inherited an X chromosome from their mother and they inherited a Y chromosome from their father. And look at this X and Y chromosome down here, and the male karyotype here. Notice that the Y chromosome is not homologous to the X chromosome. You can see that it's a much different size. And so we can say that human females have 23 pairs of homologous chromosomes because we have the two X chromosomes included, whereas males have 22 pairs of homologous chromosomes, and then they have an X and a Y chromosome. So meiosis happens in germ cells, and germ cells are the cells that are going to divide into either egg or sperm cells. And they start out with a diploid number, so just like the rest of your body cells, germ cells are going to have one copy of each chromosome from your mother and one copy of each chromosome from your father. And so here the cell starts out diploid, and one of each of these pairs of chromosomes is going to end up in the cell that will become either the egg cell or the sperm cell. And meiosis is the process that determines which of these chromosomes end up in which cells that are produced. So both egg cells and sperm cells are what we call haploid cells. And haploid is represented with this symbol N. So a haploid cell is only going to have one of each type of chromosome. And then when an egg cell and a sperm cell fuse together, we say that the diploid number, right, this 2n number, or 2 of each type of chromosome, has been restored. So this slide just kind of shows the same thing, right, that the goals of meiosis and sexual reproduction are to reduce the number of chromosomes. So if both parents start with 2 of every chromosome, by the end of meiosis, the different steps that happen, we want to end up with 1 of every chromosome in each of the gametes that are made. And then sexual reproduction does the opposite. When sexual reproduction occurs, a cell from the female fuses with a cell from the male, a gamete from a male, and forms what's called a zygote. And a zygote is basically a fertilized egg cell, and it's going to go back to having that diploid number of chromosomes again. We would also call this genetic recombination, because we are recombining genes from the mother with genes from the father in a new way in which we could end up with novel combinations of traits that didn't exist before. So let's look at the process of meiosis in general. Comparing meiosis to mitosis is pretty easy to do since we just learned about mitosis. But in meiosis, there are 2 rounds of divisions, not just one, like in mitosis. In meiosis 1, the homologous chromosomes are going to pair up with each other. When they do, it's called synapsis, and then they get pulled apart from each other. This is very different from what happens in mitosis. But then in meiosis 2, the sister chromatids of the duplicated chromosomes are pulled apart, and we end up with unduplicated chromosomes. This is actually very similar to what we see in mitosis. So let's go through our visual tour of meiosis. Starting in prophase 1, when the homologous chromosomes pair up and synapsis happens. So think of synapsis as the process in which homologous partners have paired up and they're touching each other in places. So here are 2 duplicated chromosomes that are homologous to each other in synapsis. Here are 2 more duplicated chromosomes. We can also see the other things that are typical of prophase, right? The nuclear membrane is breaking down. The centrioles are starting to move to opposite sides of the cell. So there are some similarities here between prophase of mitosis that we just learned about. Moving on to metaphase 1. Here is where we're going to see a very big difference between metaphase 1 of meiosis and metaphase of mitosis. Because if you remember in mitosis, if we had 4 chromosomes, they would all be lined up in the middle in one line. But this is meiosis 1, and so instead the homologous pairs line up. So I've got a pair here and I've got a pair here. And that's because what happens here is the homologous pairs get separated from each other. Because remember, ultimately we want to end up with half of the chromosome number, and this is the first step to make that happen. So here they get pulled apart during anaphase, and now, even though we started with a diploid number of chromosomes, right, 2 of every type of chromosome, now already at the end of meiosis 1 in telophase 1, we've already got a haploid number in these new cells that are starting to divide from each other. Because I have 1 of this small type and I have 1 of this large type. And over here I have 1 of the small type and 1 of the large type as well. But I started with 2 of the small type and 2 of the large type in this cell. So let's go on and look at what happens in meiosis 2. So here in meiosis 2, now I've got these 2 cells that are already haploid, right, because they have 1 of each type of chromosome instead of 2 of each type of chromosome. And they're going to go through a process that looks very similar to the process of mitosis. So they're going to line up in the middle in metaphase 2. The sister chromatids are going to get pulled apart and become single unduplicated chromosomes. And notice that we started with 2 chromosomes that are duplicated. We ended with 2 chromosomes that are unduplicated. Okay, so the chromosome number hasn't changed, just whether it's in its duplicated state or its unduplicated state. So in the end, after these 2 rounds of division, notice that we have 4 haploid cells. They each have half the number of chromosomes than the original cell started with. The other thing I want to point out that's really important is that these haploid cells have different combinations of chromosomes, right? So let's say that this light blue color came from the paternal chromosome and the dark blue color came from the maternal chromosome. So in this cell, there was a big chromosome from the dad and a smaller chromosome from the mom. And in this cell right here, also had a big chromosome from the dad, but there's a little piece from the mother's big chromosome, which happened during prophase 1 of meiosis, during this period where the chromosomes were touching each other in synapsis. And what happens here is that little pieces of the chromosomes interchange with each other where they're touching. And we call that crossing over, and that's a pretty important vocabulary word. So in the end here, the chromosomes have been changed. Some of them have a mixture of the maternal genes and the paternal genes. And remember that a gene has the same locus on every chromosome, and so when a little piece of the maternal and paternal chromosome got swapped here, the chromosome still has the same genes, but now it's not all genes from the father. It's a mixture of genes from the mother and the father. And then down here we see the same thing, a mixture of genes from the mother and the father. And so in 4 different cells, we have the same genes, but we have different combinations depending on which chromosomes ended up together and depending on crossing over and how that happened as well. So this is super important, right? That you guys notice that these are genetically different from each other. This is very different than what happens in mitosis, because remember, in mitosis, we want to end up with identical copies of cells from the parent cell. Not in meiosis. We want the different cells to be genetically different from each other. So let's talk a little bit more about this process of crossing over. So as I mentioned before, it happens during prophase. When the 2 chromosomes are in synapsis, when they're touching like that, we also say that they're in their tetrad arrangement, because you've got 2 duplicated chromosomes, meaning there's 4 sister chromatids there. That's where the tetrad word comes from. There's also a process that happens during meiosis that is called independent assortment. And this is an incredibly important process as well. All right, so let's say I have an organism that has 3 pairs of chromosomes, normally, in its diploid cells. Well, that means its gamete is going to have 3 chromosomes. So here we've got a paternal gamete, this would be a sperm cell, and over here we've got a maternal gamete, which would be an egg cell. And when they fuse together, we get a diploid cell, right? So now it has 1 of each type of chromosome, and since there's 3 types of chromosomes, that is 6 in this case. This organism right here, when it's ready to make its own gametes, there are various ways in which the paternal and the maternal chromosomes could combine with each other. So you see this pink color is representing the paternal chromosomes and the green color is representing the maternal chromosomes. So you could end up with all paternal of each type. You could end up with these 2 paternal and this 1 maternal. You could end up with these 2 paternal and this 1 maternal, right? So these are just showing the different ways in which these different chromosomes could be combined with each other to end up in a single gamete, because each gamete is going to have 1 of each type of chromosome. And we call this independent assortment because how the chromosomes are paired with each other is independent, right? Not all the maternal chromosomes are attached together, not all the paternal chromosomes are attached together. They are randomly sorted and could end up with any of the other chromosomes, and that's why we call it independent assortment. So here's another way to look at independent assortment. So once again, let's talk about an organism that has 3 pairs of chromosomes in its diploid cells. So During metaphase 1, those chromosomes could line up like this, or they could line up like this, or they could line up like this, or they could line up like this. So depending on which way those chromosomes line up, and this will start during prophase 1, because prophase 1 is when the spindle fibers actually start to attach to the chromosomes, and depending on which spindle fibers attach to which homologous chromosome is going to determine how they line up in metaphase 1. So during prophase 1 and metaphase 1 is the important time that's going to determine how independent assortment will occur in this cell as the gametes are being produced. So then if we look at metaphase 2, in this example, here's what we would get, right? And if we go down, we see these different combinations ending up in the new cells. And then by the time we get to the end of meiosis, here are all my different genetic combinations of gametes based on how this occurs during prophase 1 and metaphase 1, right, during independent assortment. And so in a cell like this, where there's 6 pairs of chromosomes to start with, so there's 3 pairs of chromosomes, each with 2 possible ways to line up. So this gives us 2 to the 3rd, or 8 possible different nuclei in the end, if we count these. But in humans, because we have 23 pairs of chromosomes instead of 3 pairs of chromosomes, then that means we have 2 to the 23rd possible combinations of homologous chromosomes. Right? So that ends up with this huge number of different possibilities of paternal and maternal chromosomes ending up in a single gamete from a single individual. And so combine this with crossing over, and now we're looking at an even bigger number than this, right? And this would explain why your siblings don't look exactly like you unless you're an identical twin, because the likelihood of getting the exact same possibility again from both parents and both of those combinations combining with each other during sexual reproduction is so unlikely that we do have differences. So between crossing over and independent assortment, therefore, what we end up with are novel combinations of alleles, right? So lots and lots and lots of new traits and new combinations of traits that are going to appear among offspring. And this is something that is unique to sexual reproduction. All right, so just a quick mentioning of how gamete formation in males happens differently than in females. So don't worry so much about all these terms, primary spermatocyte, primary oocyte, but we call gamete formation spermatogenesis and oogenesis in females. And so meiosis happens the same in both males and females, right? Because remember, meiosis is going to be how the nucleus is dividing. But what happens differently in males and females is that the cytoplasm divides differently in females, whereas in males, the cytoplasm divides evenly. So at the end of meiosis in males, they end up with 4 sperm cells that are each haploid cells. But in females, that first round of division happens unequally when the cytoplasm divides. And so you get one cell that has most of the cytoplasm, and the other cell just has a little bit of cytoplasm. We call that a polar body. And its main purpose is to kind of take away those extra chromosomes and get rid of them, because we want to end up with an egg cell that has a haploid number of chromosomes. And so these 2 rounds of division happen in order to separate the nucleus appropriately so we get a haploid number, but cytoplasmic division happens as well. And so after the first polar body is formed, that secondary oocyte, or the bigger cell that had most of the cytoplasm, divides again. And then there's unequal cytoplasmic division again, and we get another polar body. So in the end, females during gamete formation end up with one ovum that has a haploid number, And then they end up with these extrapolar bodies, which just degenerate. They don't really serve a purpose other than to get rid of those extra chromosomes. So this is a little bit different than how it happens in males. And if you think about the evolutionary purpose of this, these male gametes, their main function is to swim real fast and carry their chromosomes and fertilize the egg cell, and that's all they really need to do. This ovum has a lot more responsibilities. It has to start with all these extra materials, and it has to be ready for this cell to start dividing and start supporting what is going to become the actual organism. And so it would make sense that cytoplasmic division between the two different genders is a little bit different, right? So once again, this is oogenesis versus spermatogenesis. All right, so we've been spending quite a bit of time talking about sexual reproduction. Let's look real quickly at asexual reproduction. Right? You've heard of this before, and asexual reproduction requires one parent. And so there's various ways that asexual reproduction can occur. Most of us think of binary fission. When we think of asexual reproduction, we think of a single-celled organism that splits into two. But asexual reproduction can also occur in a few other ways, right? Some organisms can bud, right? So a little piece of the parent organism can bud off into another piece, and then this can grow into its own new organism. We see plants doing the same thing when we take a cutting from a plant, for example, and plant it somewhere else. That's a form of asexual reproduction. Some organisms can regenerate from fragmentation. So there's various forms of asexual reproduction, but the point of asexual reproduction is that it's fast. It's a very quick and efficient way to produce more offspring. But there's very little variation, right? Variation is introduced through mutations, almost completely through mutations. Whereas in sexual reproduction, variation is introduced through mutation, but also this genetic recombination that we just talked about. The genes from mom recombining with genes from dad, and you end up with these very different combinations of traits. This does happen at a slower rate, though, right? So sexual reproduction is much slower than asexual reproduction, and it requires a lot more energy. So think about all the mating rituals out there. Males fighting over females, or males trying to impress females, or females trying to impress males, right? There's a lot of energy that goes into the rituals that determine how sexual reproduction occurs. And so this has to tell us that sexual reproduction being slower, that energy is expended on mating, right? There must be a reason why this is such an advantage over asexual reproduction, because otherwise, why wouldn't everybody just grow a new individual by taking a little piece off of their thumb and growing a new person from it? Like, if that's a quick and easy way to do it, then why doesn't every organism just do it this way? Well, as we discussed already, the result of sexual reproduction is to get these genetic recombinations. And so what you end up with are a lot more genetic variety. You end up with offspring that are not identical to each other, that are not identical to their parents. And this provides a lot of raw material for natural selection to occur. Because if you have a changing environment and you don't have enough variety in a population, then if there aren't traits in that population in which they can survive through the changes in the environment, then that whole population is going to die. So having genetic variety has been such an advantage over time that sexual reproduction has been so successful, even though it requires a lot more energy than asexual reproduction does.